So our first speaker is Mitch Stanton in chemistry. Mitch completed a Bachelor of Science honors degree at BU with a major in chemistry and a minor in biology, and you finished in April, I think,
Yeah.
of this year. Yeah. And he'll be speaking to us on NHC Carbenes as biosensors. So presenters have about 15 minutes with some time for questions. I might limit questions a little bit and just to try to get us a little bit back on track, but I also don't want to take away from the students either. So we'll be trying to stick to the schedule, but being reasonable at the same time. So take it away, Mitch.
Sweet. Thanks for the introduction. Thanks everybody for coming to listen about what I'm going to be researching for the next 2 years here at Brandon University. So what exactly are NHC carbenes and heterocyclic carbenes? They are a 5 or 6 member cyclic system. Generally, they have 2 to 3 heteroatoms like oxygen, nitrogen, sulfur, something like that. They do have quite a few different applications. One of the big ones is that they are catalysts in organic chemistry, which is kind of interesting. They act as ligands in organometallic chemistry. They actually also have a role in antibiotics as well, which is pretty cool, pretty interesting. So same thing, they've just been kind of recently starting to be explored as biosensors for detection of proteins or chemicals, that kind of stuff. So, but as you can see, that's kind of like the general structure of them, 5-member ring. There's also derivatives of them where it still has the same imidazole backbone. So just to kind of a couple examples. So this is them being used as an organic catalyst so all the actual chemistry kind of happens right where the red dot is. So I had the actually honor to synthesize and use these last year in my honors, which was really nice. Other cool thing about them is in 2021 they won the Nobel Prize in Chemistry for their discovery and use of inorganic synthesis. So other thing too is they also are used as in organic or organometallic chemistry. They're ligands, just as you can see, that still has kind of the same backbone, the same imidazole derivatives. So other cool thing, they actually also have been starting to be researched a lot in anti-cancer therapy in terms of they cause cell arrest, anti-cell proliferation, sorry, and so they do have kind of like a pretty wide range of applications they can be used on. So, but as biosensors, it's kind of what I'm going to start looking at with them. So what exactly is a biosensor? It's an analytical device. It's used for detecting proteins, bacteria, or some kind of chemical that you're looking for in a solution. They can be used for qualitative and quantitative results, which is kind of handy. And then just a super like basic diagram of how they work. You would have your analytes. That would be like what you're looking for in your solution. And then you have your bioreceptor. That's kind of what I'm looking at making, which is what your protein would attach to. And then the interface would be like what it's actually anchored to. And then you have your transducer. It takes all the signals and then produces it hopefully into a nice usable piece of information for you for your test. So I'm going to give you kind of a pretty good little analogy that I had explained to me from my prof to kind of explain this, how it works. So imagine you're this guy and everybody here is fished or knows about fishing of some kind, I hope. So imagine you're the guy that's you're in the fishing boat and you're hanging on to your fishing rod. You have your fishing line and then you have your hook, and your whole goal is to try and catch a fish, right? So the same thing can basically be applied to, in a really simplified version, to what I'm trying to do. So that on the right, actually, my directions are backwards, is the molecule that I'm going to be looking at. It's a benzimidazole derivative. So essentially, you can think of it kind of like somebody that's going fishing, where the probe that you would have that would catch your bacteria, protein, something that you're looking for would be like the hook to catch your fish, and then you would have your line, just like the fishing rod, which is the chain coming off the actual body of the molecule. and then like the rod, which would be the actual body of what I'm making, and then it would be anchored to gold, which would be like the guy hanging on to the gold. And essentially, so it's like the best way to think about it is like a molecular fishing line is kind of what we're making or trying to work on, which is a really nice analogy I had explained to me for this, or a comparison. So previously kind of what existed on the market, or like in us, that's been used were thiol-based biosensors. So it was just a single gold atom that bonds to gold. That sulfur has a really high affinity for gold as well. They also have this other really kind of cool characteristic to them where they make something that's called a self-assembling monolayer, where as you can see in the picture, they'll when you add them to the gold, they'll actually kind of stack themselves in all a really nice orientation and really tight together, which is a really important characteristic for us. But there's kind of a couple drawbacks to them. It's just the only thing. They are sensitive to pH change, sensitive to light, air, just generally like degrading over time fairly quick. So that kind of, and they're a little bit more expensive to synthesize as well. And then they don't have a really long shelf life either once they've been used. So with that in mind, so we're going to kind of explore those sensors, but now with the N-heterocyclic carbenes. So it's the same thing. It's just but instead of being sulfur, it's a carbon that's bonded to your gold monolayer because it has a super high affinity for gold as well, which is nice. But they also share the same characteristics as the sulfur-based sensors, where they will stack themselves in a nice uniform pattern to make another SAM or self-assembling monolayer, which is a really handy characteristic. So the nice things about these is they're a little bit more robust, essentially, is the best way to explain it. They're less susceptible to pH change, to light, to solvents, even just general degradation. They have a lot longer of a shelf life where they won't degrade. It's also a stronger bond to the gold than what the sulfur is, which helps with that sort of stability and ruggedness. And you can actually manipulate like essentially like the fishing rod of the molecule to influence and manipulate it as you want. And I'll show you on the next slide here. So, because the thing is this is kind of how they stack with each other, but those R groups, so you can change those and put really big substituents or small substituents on them. And what that does is that actually affects the angle at what it bonds onto the gold. So if you have like a really small substituent, it's gonna like lay flat on the gold, and then it's not gonna be able to kind of line itself up in a nice self-assembling monolayer. But if you have big substituents on it, then it'll make it so they stand a lot more upright and a lot more uniform, which makes it so you can have like a nicer monolayer with more substituents on it. So I've been talking about gold this whole time, like just, why not use something else like platinum or something that's like a different, or copper, those kind of things. So reason being is in organic chemistry, or in inorganic chemistry, organometallic chemistry, there's a theory that exists. It's called hard, soft, acid, base theory. And I have kind of a funny little analogy that a prof explained to me. It's, imagine you are trying to play hardball, but you're using a soft baseball. If you try to hit it, it's not going to have the same kind of connection, right? But if you play hardball, but you use the hard baseball bat and then the hard ball, then when you connect it, it's going to make a way stronger connection, right? And it's kind of the same thing with that theory, where the hard acid will only react with the hard base, and then the soft acid will only react with the soft base. And being that gold and the NHC carbene, that they're both soft, they have a really high affinity for each other to kind of make a really strong bond, which is nice. So continuing on, this is kind of getting more into what I'm going to be doing now. What we want to do is synthesize the actual molecule, the benzimidazole derivative from previously reported literature and then kind of modify it to what we want to do. And then we'll synthesize that and then basically kind of test how the efficacy or the affinity and efficacy for how it attaches to the gold monolayer, which is kind of interesting. So again, going back to like that fishing example that I had, just with the molecule itself, like the probe that we have on the top, which is going to grab your analyte that you're looking at, your sample, which would be like your fishing hook, because there's different fishing hooks for different kind of fish, kind of the same thing here. And then you have different line lengths as well, like because if you want to go fishing in, if you want to go fishing in a lake and you go fishing with a line that's meant for an ocean, you're going to go to the bottom, you won't catch anything, so you kind of want to have that right length for what you're looking for. Same thing, the rod, which would be like the same thing as the rod, it would be like the body of our molecule, and then the gold would be like the anchor, the guy hanging onto it, and that's what's holding the carbene to the gold. So one of the things we want to kind of check this first is to see how the different alkyl chain lengths will kind of affect it, if it'll affect how it binds to the gold monolayer, and also how it will, and if a longer chain will actually grab like your protein or your analyte or whatever you're looking for better or worse, which will be kind of interesting to see how it bonds to the gold, because theory is that a longer chain will actually be kind of worse than a shorter chain, but we kind of want to try something with that just to see how it works. And then once we actually get it to bind to gold, show that it binds to the gold, makes the monolayer, the next thing we want to do is attach a previously reported hook, like some kind of either antibody or a different system to prove that this works and show that it works. And from there, then we actually want to, once we prove it works, design and synthesize our own kind of specific hook for what we want to do, or for what we're looking for. So just a couple of examples of a different kind of hooks that are already out there. There's something like this guy here. It attaches to attaches to sulfur on cysteine groups. Same thing as this guy, that's melamide. That's kind of what we're looking at right now. This also attaches to sulfur groups on cysteines, which is nice. And then this guy, which is just a different one, it attaches a little differently. It attaches to the N terminus of a protein, which is an amine group instead of a sulfur group. So, and this is where it gets kind of a little bit more into it. So this molecule here is kind of what we're basing our fishing hook off of. To kind of explain it, is this is something that's used quite often in peptide synthesis. It's called FMOC. It's a fluorescent molecule, so when you're making your peptide, you can kind of follow the fluorescence of it. We're wanting to modify it and create this guy here that would be attached to our fishing line that we have, and this will essentially should bind with either a sulfur group, or also you can use it to attach another hook. So that would be like our molecule that we have, and then from there you could actually attach an antibody if you wanted of interest. And then the cool thing though is the next part is you could attach your protein and then quantify it, but then you could react it with a super mild base, and then it'll basically break off and detach as this molecule here. Now, the reason that's kind of interesting is because this is a very fluorescent molecule. It's called a fluorophore. It's meant for tagging and labeling molecules with a fluorescence, essentially. So, but the other kind of interesting thing that's more cool, in my opinion, is that once this breaks off, this will decarboxylate, let off CO2 gas, but it'll leave us that amine molecule there. So what I want to try and do is basically have your self-assembling monolayer and then reattach a probe back to it. So instead of having to use like a new probe every single time with the antibody on it, then you could just reverse the reaction, attach your probe and antibody, and then have a analytical tool you can just kind of reuse. So and then in terms of future directions, First things first, I need to finish synthesizing the molecule, and then attach a known hook that's like a known fishing hook, or that we know that works in literature. And then after that, attach our own hook and try that out, see how it works. And then kind of also, while we're doing that, compare the alkyl chain lengths to see if that influences it whatsoever. So, and then just a thank you to my sponsors, funding, and my advisory committee, and Department of Chemistry for letting me use their equipment. And thanks you for listening to my presentation. I'd be glad to take any questions.
Okay, Dr. Malcolm will take over emceeing for the rest. Oh,
I was going to do yours, but I can do all.
oh, sure. Okay, I'll do the first half and then you do the second half. Yeah, perfect. Okay. Right. Any questions for Mitch? One or two quick questions so we can try to get back on.
I knew you were going to ask a question, so
get back on track.
I was just
preferably a yes or no answer question.
Yeah, make it just a yes or
no.
No,
I'm just teasing.
Simple, please.
Multiple choice is preferred.
Mitch, just want to know, because you did mention that you are trying to develop it. Like, what are the major hurdles you faced so far?
So the first one that I have faced so far is actually the synthesis of the starting molecule. It's called 4-amino-3 nitrophenol, and I had a tough time synthesizing that. And now that I have that synthesized, that everything else is actually kind of going smooth. And yeah, basically what it was is on the ring, I had oxygen somewhere, and it kind of directed it to where I didn't want the nitro group to go. So then I had to attach a substituent on the oxygen to deactivate it to make it so the nitro goes where it's supposed to. So then I can have the amine group and the nitro group beside each other to kind of cyclize and make the benzimidazole. That's the hardest thing that I've done so far, or the biggest problem that I've had so far.
Just a conceptual question. I am a neuroscientist with a modest knowledge in chemistry,
Okay.
but with a strong interest in biosensors.
Okay.
So in your definition of biosensor, I wonder whether a electrode or a chest here,
Yeah.
would it fall within the category of biosensor given your definition, or?
Say that. Oh, like would this fall in,
Yeah.
like in terms of like a biosensor? So like
Yeah, exactly. The
yeah.
definition you gave at the-
Yeah,
the
because
simple electrode. Yeah.
yeah, if you had
Yeah, chest electrode when you are jogging, for example.
like an electrode that would be able to detect
Like
it
a
on the fly.
Fitbit that monitors your heart rate. Is that a biosensor?
Given that definition.
I guess technically speaking, yes, I would think so. Yeah,
Thank you.
I hope that answers. Yeah, sorry, I was not expecting a question quite similar to that. No, that's great. Thank you.
Okay, we will move on to our second speaker. Okay, our second speaker is Anonna, and she completed her master's in clinical pharmacology or clinical pharmacy and pharmacology in Bangladesh. And she's also worked as a hospital pharmacist in the United Arab Emirates. Go ahead, Anonna.
Thanks, Pete, and thank you all for being here in our talk. I brought my master's proposal to share with you, which is investigating the role of TXMDC12 in breast cancer. First of all, breast cancer is the abnormal growth of breast cells, which is the second leading cause of death in Canadian women. But the good part is 5-year survival rate of breast cancer patient can be improved to 99% only by detecting breast cancer early. That means we need more biomarkers for detecting breast cancer. Our laboratory identified such biomarkers which can be used for detecting breast cancer at very early stage, which are microRNAs or miRNAs. What are these miRNAs? These are small non-coding endogenous RNAs. From central dogma of life, we know that the recipe of making protein, which are the main functional molecule in our cell, is from DNA to messenger RNA, and messenger RNA code for protein. microRNAs are also RNA. They also come from DNA, but they do not code for protein. Instead, they work by targeting this messenger RNA and stops making formation protein, which is a crucial part of gene expression. And this dysregulation can lead to the tumor growth in different kind of cancer. Now, our laboratory works with two specific microRNA 526B and microRNA 6x5. It has been shown that these two microRNA high cells encourage the growth of tumor phenotype or tumoric properties in cell when overexpressed, meaning when there are more RNA copies of these RNAs, they show this phenotype. What are the phenotypes? First one is oxidative stress or increased reactive oxygen species. They gain hypoxic response, meaning when there is no oxygen, cells want to die. And migration and invasiveness, meaning normally cells are static, they do not want to move. But when they gain tumoric phenotype, they move faster and invade. That is called, they migrate and invade, spread faster. That's called migration and invasiveness. Now, not only these microRNA high cells, when we collect the secretion, whatever they're releasing in the surrounding, altogether we call it cell secretome. It has been shown that the cell secretion, if we collect and feed to the normal cell, which are non-tumoric, they also gain the same phenotypes, these all phenotypes. That's what made us think there might be something these microRNAs are releasing, what is regulating the tumor surrounding or tumor microenvironment. Now, this was a project of one of our ex-master student, Riley Facer. What he did, he collected the cell secretion and he analyzed this by mass spectroscopy to identify whatever the secretory molecules available there. Now, this is the result you are looking at, the cell secretome analysis. He identified 1,535 molecules, and after applying different bioinformatic tools specific to breast cancer, he cut down the number to 34 secretory molecules, which are encoded by 8 specific genes. And you see in the heat map, these 8 genes, top 4 in the red are upregulated, means they work more, and down 4 in the blue are downregulated, means these genes work less. Now, I'm particularly interested about TXNDC12 to know that what TXNDC12 is doing in the secretion in the first place. Now, what kind of protein is this? This is a thyroidexin family member, meaning structurally it is, as this name suggests, thyroidexin domain containing protein 12, big name. Now, this is this functions basically by maintaining the balance between oxidized molecule and reduced molecule in cell, which is called redox process. Where can you look at them? Mainly it's found in endoplasmic reticulum intracellularly and also in extracellular space, means it's more available to function. What normal function of TXNDC12, as I mentioned earlier, maintaining the redox balance, also maintain the cellular stress response, helps the protein to bind or form, and they are associated with different kind of neurodegenerative disease and also cancer, also take part in intracellular signaling. Now, as I said, I was curious what TXNDC12 is doing in the secretion, and I was also thinking how these two microRNA are regulating TXNDC12. What is the regulatory connection? In our cell, there are thousands of molecules who have their own regulatory formula, or they have the regulation process by targeting specific molecules, kind of superheroes who follows their instruction from the gene or the secretory molecules. These are called transcription factors. TXNDC12 has 33 transcription factors, and when 2 microRNA common targets brought together, we find 4 common targets, meaning amongst these 33 transcription factors, we have 4 transcription factors which are being targeted by these 2 microRNA as well TXNDC12. Now, what are the transcription factors? KLF10 and NO are the 2 negatively regulated transcription factor, and IKZF3 and MYC are positively regulated transcription factors. Negative regulation means they actually put some brake or restrict the gene from functioning, and positive regulator helps them to work faster. Now, coming to the gene expression of TXNDC12 in patient tissue, as you can see, TXNDC12 gene expression was upregulated in tumor tissue in red compared to the normal tissue, and each dot indicating the number of samples, what is mentioned on the left side. And from the right Kaplan-Meier survival plot, you see high expression of TXNDC12 is significantly connected with poor patient survival, meaning the patient who has high TXNDC12 expression are going to die early. Here you see the pink line showing the high expression of TXNDC12 it is having less number of dots, which indicates the survival probability in the y-axis, which is mentioned in the y-axis, and x-axis is showing you the number of years. Now, Riley, what he did, he validated all these data in microRNA high cell secretion, where he found this all, upregulated also. And he also validated this in different breast cancer cell line. And TXNDC12 expression was found significantly upregulated in T47D and SKBR3. This is microRNA low epithelial cell line and HER2 positive cell line compared to MCF710A, which is non-tumor, used as control. Now, after studying all this, a few questions actually led me to ask this question, which are indeed my research question. First of all, does TXNDC12 play any role in oxidative stress? If yes, then how? And if this is directly involved in the tumoric phenotype change as a consequence of oxidative stress, if yes, then how? As I mentioned in my second slide about the tumoric phenotype. So objective number 1 is to investigate the effect of TXNDC12 in TXNDC12 knockdown in reactive oxygen species. For doing that, I'll be taking my experimental cell line, which are already high in TXNDC12. I'll be knocking down them by using siRNA, meaning I'll be reducing their gene expression, and we'll be measuring the gene expression after knocking down, also protein expression, and I'll be measuring reactive oxygen species and superoxides. And after that, I'll be treating the experimental knockdown cell lines with hydrogen peroxide to induce oxidative stress. I'll repeat the process and compare the results. And I'll also analyze the gene expression for nanogen calf10, as I said, 2 negatively regulated transcription factor. Now, this method will tell us if TXNDC12 is actually involved in oxidative stress, and if it is targeting these 2 negative regulatory transcription factor or not. Coming to objective 2, which is to identify the role of this gene in tumor phenotype change. Now, I'll be doing functional assays before and after knocking down this TXNDC12. Functional assays like migration, invasion, and proliferation assays. And then I'll be analyzing the EMT markers, meaning epithelial to mesenchymal transition markers, which are the responsible molecule for this phenotype change. And I'll be also knocking down one of the markers, ZEB1. Now, this method will tell us that if TXNDC12 is changing the tumoric phenotype, if this is targeting ZEB1 or not. Now, to start with my first objective, to knock down TXNDC12, I needed protein expression data, and my bioinformatic analysis didn't show any available data or yet reported. So I did my protein expression of TXNDC12 in different breast cancer cell line. The image of Western blot, you can see the differential expression for different breast cancer cell line. And from the quantification, we can say that TXNDC12 was high upregulated in 2 microRNA high cell line, MCF756B and 655, and also in T47D, MDMB231 and SKBR3, compared to MCF parental, which was the control. As I said, our laboratory identified 2 biomarkers, primicroRNA 526B and primicroRNA 655. So it is essential to analyze the gene expression of this in our experimental cell lines. And I found that primicroRNA 526B was upregulated in amongst these in my experimental cell line MCF756B compared to MCF7 parental, which was control. And for primicroRNA 655 also, it was upregulated in microRNA 655, also in MCF7 COX2 and MDMB231 compared to MCF7 parental, which was control. These 2 are my positive control for both of the experiment. Now, after coming to the future goal, after completion of my 2 objectives, I would like to see if there is any contribution of TXNDC12 in DNA damage, as this could be a consequence of oxidative stress. And also after that, I'd like to validate all my expression, like gene expression and protein expression in patient specimen, meaning tumor plasma and tumor tissue. And after, I expect that maybe after completion of the whole project, this would identify the role of TXNDC12 in breast cancer as, in oxidative stress. and it could be a potential tumor marker for oxidative stress responses and can help the physician to decide about the targeted therapy. With that, I'd like to thank my supervisor, first of all, for answering me at 4 a.m. and all my colleagues, super supportive colleagues, and my committee members, my 662 friend and instructors, Pam and Pete, and my collaborator and sponsor. And at last but not the least, all of you for listening to my long breath and my project. Thank you.
Anonna, when we are talking about a lot about oxidative stress, I think audience should know what it means, why it is so important to know, or having a marker to detect that.
Okay, so oxidative stress means, as I said, we have a redox process ongoing in our body and cells, meaning that there are oxidized molecule and reduced molecule. If this process is imbalanced, we get, redox process imbalanced. This results to increased number of reactive oxygen species. This, as a consequence, put the cells in stress, under stress. This is called oxidative stress. Now, what was your question? If why I do want TXNDC12 to play a role in oxidative stress? Because its main function as a gene or a molecule, its main function is to maintain the balance in redox process. So that's why I was thinking it might have any role in microRNA background in breast cancer.
No.
So in our bodies, don't we have things like superoxide dismutase and stuff like that that
Yeah.
takes care of reactive oxygen species?
Sorry?
So our bodies have inbuilt mechanisms to deal with reactive oxygen species, right?
Yes, we have our autoimmune process, but still, suppose when we have infection and it's beyond the capacity of our WBC, we still get the infection.
Okay, so
Our
therefore-
WBC is fighting us, but
No,
still we get the disease.
completely understand. So therefore, a stupid question comes next: is how do these systems understand when it's beyond the threshold of our bodies to combat it by itself, or
Okay,
by the superoxide enzymes that get rid of oxygen species?
Okay, okay. If I got your question, how our body recognize that this is beyond the
Correct.
tolerable limit?
Correct.
This is also a part of immune process, autoimmune process. Like until a specific level, our WBC can give the signals and our other secretory molecules or the transcription factors can target the infection or the whatever the problem is there and can neutralize. But after a specific level, body itself shows or says that to our WBC that we cannot deal with that more, which shows different kind of characteristics, or I'd say, yeah, what tells here comes the diagnosis. When you're feeling some kind of problem from your body, then you know that you are having problem and you go to the physician and they diagnose you with these characteristics or the properties, the problem you're facing. If it answers your question, I don't know.
Yeah, it's a very good one. I'm a little.
Okay, our next speaker is Stephen Holden, who studied undergraduate computer science and biology at BU. At the intersection of these fields, his research focuses on networks in nested complex systems evolved and engineered. Through the interpretation and processing data from biological systems, we can approach understanding both machine learning and emergent information processing in living systems. Not really a bio, I think that's more of an explanation of what your talk's going to be about, but anyway, take it away, Stephen.
Hi, thanks for being here. Yeah, so I deal with a type of complex data in spatial phenome systems, and we're going to. I'm very happy with it. I'm nervous. Okay. We planted 1,000 types of barley from Anna's genetic stock, which you discussed when she was talking about breeding. And then James's disease. He has a series of leaf disease. This is a Colchibolus nevus inoculation that straw is infected and was laid down by part of the many teams that make things happen in the NHC. During harvest. So we have 1,000 types of barley here under fairly severe disease pressure, and we control the environment so that we have maximum expression of disease against these 1,200 segregating genetic varieties from Anna's stock. You can see here the characteristic lesions on the leaf. This plant is not going to make it. This plant is very susceptible, but we do have fairly resistant stock, and also here you can see guard rows, which are ripening and senescing differentially from the less susceptible stock that is under test in James's nursery. And you can see here how they're changing color. This is close to the end of the season, and we can consider how, if we look at that change over time, that we might be able to gather and cluster the varieties from the change over time. And this is the idea I'm going to really try to expand on. There's also a great degree of surveying that goes into this. This is one of about 15 to 20 ground control points that are surveyed at centimeter level accuracy. And we did a series of 10 flights, which gives us a number 2 to 3 terabytes of mapping data. So that's one of the major issues with this approach. And finally, harvesting was done under a high degree of control. We wanted to keep biomass and seed heads, so they're bagged before they go into the dryer, and they're taped so we don't lose weight, we don't lose grain. So this was about 3 weeks of fairly intensive effort, and that gives me a very large and varied ground truth data set, where we have disease resistance scores, biomass, height, categorical types from databases, stem area for normalization, and potentially yield if we choose to go and process that, and over 1 billion data points, but we could consider those as sequences, so 100 million valid training sequences for modeling on. The idea is about to get fairly abstract as we explore this type of data, but I ground it by thinking about extrusions and literally a pasta machine, because we know the location. No matter how complex the curve gets, we can route that complex curve to a space, and that spatial routing of our survey data is very crucial to exploring that. And as abstract as it could get, I think about different materials wrinkle and curve in different ways, and that's crucial to exploring this. The phenome of any given material is not represented well by a single instance of observation, because we know that these are dynamic systems that emerge at the interface of the two organisms over an extended period of time. And so we might consider how disease onset is relevant to our assessment of genetic breeding stock. I think that Anna and James made that point very well, but we can also consider the information richness of the shape of change over time in a location. And that is, we can look at that with different sensors by observing color change, or we can do that by observing health index changes and rates of photosynthesis changing over time, as opposed to looking at a single instance in time. Any picture you've ever seen is made up of layers of red, green, blue. These are individual components of a vector in a vector space. We don't often think about that, but because our survey data is rooted very accurately in space, we can look at changes in that vector space over time and model them as a sequence. The height that we flew at was the smallest we could get at a 3 millimeter pixelation. Now, often this type of mapping is done at inch, 2 inch, 20 foot pixelation size. So pixelation is a major part of this type of data, even though we don't often think about it. I have a 7-dimensional pixel. We have infrared and different health indices that we can calculate from this, and we can use this as a proxy for rate of photosynthesis or overall plant health. Here are some of my friends from the weeks I spent in the field. There's a number of ladybugs in this image. Obviously here at a 3 millimeter pixel, they're harder to identify, but the point I want to make is that in pixelation, information is not destroyed rather it's aggregated. And so even though it's hard to count the 12 ladybugs in this picture, if we had a before and after, by the shift in just the redness channel, we would be able to identify their exact location. And so by change, we can infer sub-pixel resolution details. Physiological traits, which we're sensing with reflected light data, reflected spectroscopy, is always about sub-pixel resolution. We're never able to see in a pixel the physiological phenomenon that's driving reflectance. And so by being able to look at change over time in a dimension, we can look and infer the physiological process that underlies it, even though we can't resolve it in the pixel. Recall that this is a noodle rooted in space, moving in over 7 dimensions. So we actually have a phenomenal amount of information there. How we extract that is something that I've spent the last year working on. And the simplest approach would be to take the expression of an individual location over that time series and model that equation, then do math that I need friends to help me with, take an inner product, do a comparison. But this is computationally inefficient. And also what happens in any way I tried to approach modeling these curves, which I think have a great degree of usefulness for us, we lose the time sense. We're constantly collapsing time. That sense of time is the most crucial differentiating signal and value when we're analyzing different crops. And so the transformer as a form of machine learning is well discussed, but I think the attention head has a very clear application to this type of time series. Given that I have 10 readings over 7 dimensions, if I take a reading by dimension matrix and we take the transpose of that matrix, what we get out of it is reading by reading. And we can learn the weights of the significance for each reading related to each reading. So the final value knows about, it learns the significance of the first reading. And they learn the significance of each other in their relationship by taking the weights of the transpose matrix. That final attention matrix represents an embedding of the sequence of changes in the phenomic sensor space, semantically embedded in their significances over the season, as we're looking at the interaction of barley and this pathogenic fungus. Over 1,000 varieties. But that's difficult to observe. And so this is the spicy meatball in the pasta. A beta variational autoencoder has some very special properties in that it can be used to disentangle a number of factors, and it's often used for clustering complex types of data. What it also gives us is the ability to do is once we have those clusters, we have them in their individual dimensions. It natively wants to separate the dimensions of data, and it will leave unused dimensions empty, essentially, and it will actually reveal the dimensionality that's in the data over. So maybe that's a 20-dimensional data set. In its interaction, it will leave the remainders empty, and you can sample from among those and generate synthetic data that allows you to observe changes that could be, and you can infer from that exactly how the model's learning and the meaning of the individual readings that you had at a given pixel. You can map and model their weights and visualize them. And essentially what you get from this architecture is the ability to train sequence to sequence, so you don't require any data labeling. You take just the phenomic, the transition of the phenome over the 10 readings of the season. That raw sequence has an embedding in that vector space of the sensor. The transformer contextualizes it into an overall semantic embedding. The beta variational autoencoder clusters it by the overall meaning in a disentangled way that we can explore and hypothesize and observe over. And from that, you're able to model the sequence
infer underlying phenomena from the superficial sequences. So we talked about inferring subpixel information. This is known to be a method for projecting a phenomena from its representation. And so a lot of other approaches to disease recognition and trait identification use segmentation models. They're very popular. They're very powerful. They happen to be extremely labor intensive. Labeling can take half of a PhD. And after you're circling your 100,000th instance of a lesion to train the model, people get a little bit loopy when you're 5 years into that. And once you're done, the model is really at an end state, generalizing from plant to plant and process to process and disease to disease. You put this enormous amount of labor in, and the model is very difficult to extend, if it even can be done. Whereas this approach, the beta variational autoencoder, is in fact and was designed to be a form of compression. So this takes the multi-terabyte scale of data and actually gives you a way of databasing and scaling season length data sets. It also lets you train to identify the underlying phenomena without labeled data. You can just go from sequence to sequence and model it. It takes a lot of compute power. Once you have done that foundation model over the phenomic sequences rooted to locations, then you swap out some very final components. The model is extremely shareable at that point. You share the architecture, you share the weights, and then you do a fine tuning on the end to fine tune it for application. And this can be done on a local computer with regular compute. It's much less computationally heavy to form the application. And this is because we're taking a semantic approach. This is highly shareable and extendable and can move along even much more general concepts of biological change semantics. And the implications of this, where I'd like to go with it in the end, is it's trivially easy to extend the dimensionality of a pixel in a location to incorporate environmental data or other factors. The dimensionality can be expanded to whatever compute you have available to you. In our case, we've controlled the environment, but that's not necessarily the restriction whatsoever. And additionally, model observability of the design of this architecture is, in fact, what I've designed it for. As scientists, we need to be able to know and understand the phenomena. And given a native embedding, at every point, we can understand the meaning of every pixel at every node. And so it's a general tool. Thank you.
So most of the work that you're doing is obviously like designing this model, right?
Good question.
Have you like, I guess, like tested it at all?
The model design is not the most difficult part, actually. These two models, I've just scratched the surface, but their use, application, and properties are well addressed and extremely well characterized, in that a transformer is semantic and a beta is highly clustering and observable. The most difficult part is actually data manipulation and preparation. That's very difficult. But the literature support for extracting the semantics of a vector sequence is very well established. Where I'm differing there is that I'm using the natural embedding of the type of data we're using, as opposed to arbitrary embeddings, which is typically done in linguistic processing. The embedding of this data is meaningful, and these applications are well established.
It is really, really visible. You explained that very well, that how complex your project is. In a simplistic way, to me, I'm just trying to understand. So what is the outcome of this project? Or we are trying to predict a future infection pattern depending on the genomic information? Is that what we are trying to do?
There's so much I don't have time to cover, and I wish I could. If you go to saint-even.com, I actually do say that I'm not interested in future state prediction, but rather from data, signal extraction, and multi-trait characterization. So what you can do with this is you can, from a trained model, you can look at multiple traits simultaneously because you're working from semantic signal extraction and you're not working from identifying a single trait. You're identifying patterns of trait, patterns in phenome appearance and environmental conditions. And so you can take that and you can statistically infer over terabytes of data current state in many ways.
Okay, our last speaker before we take a bit of a break is Francis, talking about analyzing plastic waste in Africa with machine learning. Francis completed undergraduate studies in electronic and computer engineering at the Namdi University in Nigeria. Francis did a graduate diploma in science education at the University of Lagos. Previous works were in GSM controlled dot matrix display microprocessors and effects of hands-on activities at the performance of students in physics at Ojo local government area of Lagos State. Take it away, Francis.
Thank you for having me. So my thesis is in analyzing plastic waste in Africa using machine learning. We know what plastic waste are. Plastic waste actually has to do with the plastic materials found in the Earth's environment, which actually cause a lot of health hazards to humans, to aquatic animals, to animals and their habitats. So in this thesis, the objective would include, the long-term objective is to develop a plastic analysis that will actually utilize machine learning. And the sub-objective would include to provide a comprehensive review of resources and the properties of plastic waste typically found in Africa, and to develop plastic classification method for easier plastic identification and the waste modeling. And the last but not the least, to review current industrial practices and the research regarding plastic waste modeling. And the outline, of course, concept framework for total plastic management. So we have what plastic pollution is, that it is accumulation of plastic objects and particles in Earth's environment. And this thesis actually has to do with, we have plastic waste all over the world, the entire world data set, but actually there is no research in Africa. The world researchers, they somehow shy away from, researching on plastic in Africa. Hence the crux of this thesis, to look at the plastic analysis in Africa using machine learning. So the statement includes, as I said, to analyze plastic waste in Africa, and of course the unavailability of the data set in Africa make it, it seems a kind of an uphill task. But actually, this thesis delves into analyzing this using the machine learning. So these are the research questions. How effective is analyzing plastic in Africa using machine learning? How effective is it? And how might machine learning be used to categorize plastic garbage for better analysis? And does plastic waste per person include in GDP of per person, per capita, and how does the approach perform against the current industrial practices as well as research advancement in modeling? So the review, Amanwa et al., she posited that approximately 500,000 cargo containers of plastic waste imported in Africa, making it the world's second, most polluted continent in the world. And we have numerous approaches, like we have the knowledge base, we have the photoluminescence and the machine learning, we have other models. Why the choice of machine learning? The database and pollution distribution were used to estimate, visualize, and the effects of plastic waste in climate change. So the methodology I have adopted for this research includes data collection. Of course, I got my data from Kaggle, I got my data from Google and the GitHub. The data preparation, I have to train the data, I have to model the data, I have to analyze the data. So the model and of course the training and the evaluation. So this is the data set from Kaggle and Open Knowledge and the sources. So I'm using two data sets. I'm using the global plastic pollution and the African cities diagnostics data sets. So these are the features of the data sets, okay? The per capita consumption, the per capita plastic waste versus the GDP of African continents, the plastic waste per capita, the global plastic production, and of course, the per capita of mismanaged waste in Africa. So the analysis includes African, according to the classification of African continents, we have 5 regions. We have the North Africa, the South Africa, the Middle Africa, East, and of course the South Africa. So we can see with North Africa, we can see the West. This is the North. The North is embedded like Egypt, Tunisia, and all that. We have the West, Central, Eastern Africa, as can be seen in this stuff. So these are the regions we have. We have 5 regions in Africa. Okay, now the modeling approach, as can be seen, this is the world GDP of continents. We have the H here, Europe, Africa, Oceanic continent, the North America. If we look at this, we notice that Africa actually has the least GDP, but yet it is the second most continent, the continent that uses plastic waste. Now, in my research question, does increase in GDP result in increase in plastic consumption? Of course, yes, as we're going to see soon. But does the GDP increase result in mismanagement of waste, plastic waste? Of course, no. We're going to see that soon. So as we can see, the distribution of the GDP per capita. So if we're analyzing this, we actually have to look at it at the GDP, the economy of the length of the economy of the continents of the world. So that will actually give us a better understanding of the disease. Look at this. In this analysis, you can see that Nigeria, you see Nigeria, Egypt, and the South Africa, even though their GDP is not as high as that of the Gabon and Libya, but these 3 countries, they have the highest plastic mismanagement in Africa. So why is it so? Because actually the GDP increases as a result of what? When you have a robust economy, okay, the GDP will increase, okay? Now, for example, in Canada, the GDP of Canada as at last year was 1.9 trillion US dollars, and the individual per capita was 54,000. And this year, it is now, 2 trillion US dollars. Individual per capita is 54. But in Africa, like in Nigeria, the GDP of Nigeria is 4 million US dollars. And individual GDP was 2,000. But yet, they have the highest plastic mismanagement. What actually causes this? It is, of course, because of, the indiscipline. There is no clear way of managing plastic waste in Africa. If you come to Canada, I will be so stupid after using that water bottle, I'll just fill it up anywhere, anyhow. It doesn't happen. There is a clear way of, disposing plastic waste. In Africa, if you come to the sand field, you should see plastic all over. In water bodies, in ocean, in lakes, and all of that, plastic waste all over. There's no particular clear way of disposing plastic waste. So when you use machine learning from the world data set, we extract the African data set, the machine learning will learn these particular regions where we have, the high rate of plastic mismanagement. And from my analysis, as I'm going to show us, so this particular graph answers the research question that says, is there correlation between the GDP consumption and the waste management? Of course, yes. As we can see, as the GDP increases, okay, the plastic waste increases by 0.1. Okay, like that. So there is actually correlation between the GDP and the plastic waste consumption. All right. So as we can see, like I said, the Libya, Egypt, and Nigeria, they are the highest consumers of plastic and they mismanage plastic. Okay, now if we look at this, you can see this is South Africa and this is Nigeria. This is Egypt. They are the highest consumers of plastic waste in Africa. Africa contributes to 15% of the global plastic waste. Out of this, Nigeria contributes about 13.75%. And Egypt contributes about 20%. And South Africa contributes 13.1%. That's actually so huge. So something needs to be done. So I use support vector machine, which gave me a better and a higher accuracy compared to other models I used. Okay, so the CatBoost, the support vector is actually a way to go. It gives a better, learning of the plastic waste in Africa. So this is me coding using the Jupyter notebook to do my analysis. Okay, I use from Anaconda, I use the Jupyter Note to analyze my data set to get the findings. It's quite, there's no time to play this in details for you to see how I carried out my analysis. Okay, so this is the countries of Africa using the Jupyter Note analysis. And of course, this software gives a better, clear vision of the analysis. And it is a work in progress. And I will delve more because there will be a clear way of identifying, modeling, and analyzing plastic waste in Africa. And this is a way to go using machine learning. So these are the books I consulted in the course of my thesis, and I acknowledge these persons, my supervisor, the faculty members, the committee members, the Rotary Club International for supporting me, and of course my colleagues in the MELS Graduate program. And thanks for listening.
Oh, thank you. Nice presentation. I have a couple of questions, just two. Promise. One, so in your table, when you showed the methods you use to feed the data, so could you go back to that slide, please? Because so I can see is pretty much supervised learning there, right?
Yeah.
Okay. Yeah. The two questions are this. One, how do you protect against overfitting with supervised learning? Okay. That's the first one. And the second, why isn't there a possibility that unsupervised learning can give you better results or variational estimation, variational bias, for example?
Thank you so much. Actually, supervised learning will give a better understanding because, support vector machine, actually, it gives more accurate, analysis because it can learn from the data. It can actually do the prediction and the help in the modeling of the data. So supervised learning poses a better, analysis than unsupervised learning. Hence the choice of a support vector machine under supervised learning. Yeah, the overfeeding. Well, like I said, this is actually the stage I am now. I've actually not delved more in that. After now, in the completing stage of the thesis, that is what I'm gonna do. Yeah, thank you.
So I think we have a question online, and then we'll go with, or do you want to go Shrin first?
I'll grab it first if I
Okay,
could.
Shrin, then
Yeah,
Masumi.
sorry. That's what. So question from Michael and comment. So I'll just read the whole thing. If your data work consider that plastic waste in Africa include those used by Africans and those imported from Europe and routed from North America, how would your model perform with respect to per capita GDP? And then another comment, more imported plastic waste go to poorer small GDP countries and those with weaker maritime security, by the way. So feel free to comment. Thank you.
Okay, thank you so much. That's a nice question. Like I said earlier, the African plastic waste, what the global researchers somehow are shining a way, so to speak, in studying the plastic waste in Africa. This is actually because of the low GDP. Now, the plastic in Africa includes, because of the temperate, the tropical nature of the region, there tends to be more consumption of plastic waste. And this also involves the imported plastic waste from other continents of the world. And so we have, a lot of plastic waste in Africa. And this also will help, okay, in trying to reduce plastic waste in Africa. Yeah, so the plastic waste actually comes not just only in Africa, but also imported, and they come from a lot of, wide places. Yeah.
Very nice talk. The following will sound like a joke, but it's actually more serious than that. I think we don't need machine learning, we need human learning. But what I'm trying to say is, could it be possible to see, like, if you could somehow, I don't know much about machine learning, actually nothing at all.
Okay.
But if you could fit in, for example, how many schools actually teach, like you said, I'm from India.
Okay.
People throw plastic bottles here and there, right? So if we could somehow factor in how many schools incorporate, educational programs to say, hey, don't throw plastic here and there, recycle, blah, 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 is in addition to looking at the effect of plastics or the correlation between GDP, is it possible to actually factor in how many schools have educational programs that teach people and see if that trains better than the GDP?
Yeah, that can be factored, but I can tell you that categorically in Africa does not exist. Even when such laws, they can just be verbal, do not displace plastic waste here and there, but actually, because of the act of indiscipline, okay, and the culture, there should be the change in attitude for that to happen. In Canada, if you come here, you see designated places to dispose this, to do that. That doesn't exist in Africa. And it is the crux of this particular thesis. By the time I go back, because I'm just starting this, it is a crusade. I'm going to work on this, not just only in Nigeria, it will cut across African continents. There should be a clear way of disposing plastic waste. Okay, and the machine learning is actually a way to go, because machine learning will help to determine the plastic waste, the better way to dispose the plastic waste. Yeah.
Masumi?
Francis, again, I want to see the light at the end of the tunnel. So after this modeling done, for instance, as you are showing, it is almost 98% accurate. So my question to you is actually two part of it. Is the 97.8% is comparing all existing global models, or it is comparing, like as you have shown us, that South Africa, Egypt, there are difference, their management skills. So the accuracy is based on the continent or global?
The accuracy is actually based on global. Okay, like I said before, we have researchers in other continents, but we don't have, or we have limited supply of that in Africa. So the accuracy is actually based on global, and that is why we're going to use it in Africa as a model for Africa.
So, and with this, we will be better managing waste, or we will actually learn more about, is it recycle or reduction in the usage? Like, What do you think? What do you want to see?
They're not necessarily about reduction in the usage, but it will help to identify the plastic waste and a better way to dispose it, to manage it. And actually, that actually has to do with change in attitude, because what causes this is not because of the increase in the GDP of Africa, mind you. It is actually because of the indiscipline and the culture. If we just use plastic, we just do it anyway, doesn't make sense. So there should be, like he actually suggested, there should be a kind of seminar, a kind of sensitization, that will help people to do things right.
So our first speaker is Collins Chilaka, Fen Restoration in Southern Manitoba, a water balance study. And Collins is currently studying here, obviously, in the Department of Geography and Environment at BU. Is a Bachelor of Science degree in Geology from Nigeria. His research focuses on peatland restoration study with an emphasis on water balance. So it's all yours.
Welcome back. So I'll be starting a trilogy of presentations on peatlands, and you'll be hearing a lot of that in the next 2 presentations after mine. My research, like the moderator said, is on fen restoration and ecotone creation in Southeastern Manitoba, and we'll be looking at the water balance approach of this study. The aim of this research is to restore a disturbed bog peatland to a fen peatland. Now, for better understanding of what my research will be on, I will be telling you about wetlands. I will be also telling you about peatlands, the kinds of peatlands that we have in Canada, and how they are disturbed as well. I will also be telling you about the methods, or rather the technique, which we want to apply in the restoration of this peatland study. So, what's a wetland? A wetland basically means land that is saturated with water all year round. And the wetlands are made up of 2 main bodies, which is the mineral wetlands and the organic wetlands. The organic wetlands are the ones that we will be focusing on because it's the peat-forming wetlands. But in general, the Canadian wetlands includes the marshes, shallow water, and the mineral swamps, while when we talk about the organic peatlands, or rather wetlands, you will be hearing more of bogs, fens, and peat swamp. Now, wetlands are very important because they play key role in sustaining the earth in general, and the benefits can range from recreational activities to helping cool down the earth in terms of taking out carbon dioxide, which is a component of greenhouse gases, taking it out from the atmosphere and putting it into the ground as a carbon sink, and also in the mitigation of flooding, which could be very disastrous. These wetlands collect these things and try to stabilize the ecosystem. In the world in general, when you talk about wetlands, 20% of these wetlands are in Canada, which means, yes, we need to be in the forefront in playing our part in the sustenance of this ecosystem. I'll be telling you about peatlands. So peatlands, which is also a subcomponent of a wetland, it arises from the accumulation of dead organic matters that can be from sphagnum mosses, other kinds of mosses, sedges, woody plants, and all these being deposited in a marsh will give rise to possibly the formation of peatlands. So with the little cartoon I have here, you will see the processes where this forms, where we have poorly drained marsh, and once the accumulation of organic matters gets deposited in them, if the deposition is outweighed, or rather, if the decomposition of this organic matter is outweighed by the deposition, then there will be little time for it to properly decompose, and you will start seeing it filling the depression where the wetland is, accumulating, and there is also a subsequent compaction of this, and where that happens, you start seeing a dome-like structure being formed because accumulation will keep happening from time to time, and this spans over thousands of years. When this happens, you now see that dome over there depicting that it has formed a pit. Originally, these marshes, the peatland starts from a marsh, then progrades into a fen where you have certain materials growing in them. And as this goes on, the fen dies, grows, dies, laying the dead matter in layers, and compaction takes place, subsequently leading to the formation of a bog peatland. And for a wetland to be classified as a peatland, it has to have peat deposits above 40 centimeters. So why do we care about peatlands? Why the interest in studying peatlands? Well, scientists have discovered that these wetlands, peatlands play important key roles in acting as a carbon sink. When you look at wetlands and the global geographical spread, wetlands occupy just 3% of the earth. When you compare that to the immense services it provides, it is able to sink about 25% of the global carbon sink. When you also compare that to the forest and other vegetation that takes this carbon sink, the peatlands with its low number is actually doing a lot of work, and they are not that much in number. Also, like I said earlier, it stores a high amount of carbon, as much as 1,000 gigatons of carbon. And when I tell you that Canada has about 25% of the global peatland spread, it means a lot to us here in the study. In peatlands, you have basically two kinds of peatlands. You have the bogs and the fen. And in this approach, you see the peatlands actually starts from a fen and then progrades into a bog peatland. And in Canada, 90% of the wetlands in Canada are basically bogs and fen. And this covers about 13% of Canada's landmass. While our province here, Manitoba, a third of the landmass is actually a peatland and all. So this is a little spread of what I'm talking about. This here, the brownish area are where there's a low probability of pits being present, while here, the other color, bluish, greenish, has about 95% of probability of pits present in them. And that's where we are, our province, Manitoba. So what's special about bogs and fen? Why am I looking at trying to restore a peatland from a bog to a fen? It's because of certain advantages the fen has over a bog, because the bog has a low nutrient content. It also has a low pH, making it acidic, about 4.5, thereabouts. And it's homogeneous in the sense that it gets its nutrition hydrologically from the atmosphere in terms of precipitation, in rain, snow, and water vapor and the like. But the fen is the opposite in the sense that it is rich in nutrients. It also has a variable pH from above 4.5 to 7, making it basic. And it gets its hydrological feed from surface runoffs, from the ground upwelling, and also when the water table is trying to rise above to get to where the bog is and could not due to the dome shape, it spreads back into the where you have the fen. So it has a complicated hydrology, and it's more advantageous than restoring to a bog. So with these benefits, or rather with this advantage, peat bogs are, sorry, I said peat bogs, peatlands are disturbed, and there are many ways it can be disturbed. It can either be from climate change disturbances in terms of wildfires and all, and it can also be disturbed through extraction of minerals from it, oil and gas, and urbanization, where roads are being built, extraction for agriculture, forestry, deforestation, and the likes. So these are the different steps with which peatlands can be disturbed, where a peatland is identified. A machine goes in there and dig a ditch to drain off the water on it. And another goes in there to cut out all the shrubs and trees for more machines to go in with extraction. This machine actually is campers the ground, giving it a dome-like shape so that other water bodies can flow out of it into the ditch, while this breaks the surface down in case any trapped water still there can be loosened up and evaporates for the peatlands to dry out properly. And this guy goes in there and sucks up the dry peat and taking it to where it's bagged and sent to where it will now be transported out. This is the process I just described and in a much more better picture. This is what a peatland looks like when it has been impacted. It's not a pretty sight. And you can imagine all the carbon that will have been released from this site. Now, when a peatland is disturbed like this, it doesn't grow back on its own. It has a very slow revegetation state, and active restoration has to take place for this to actually be restored to the probability of a carbon sink medium. Fortunately, a technique has been designed where you can restore a peatland to a bog-based peatland. And this was designed by Lane Rochet from the University of Laval in Quebec. And this design is specifically for peatlands that have been excavated for agricultural purposes. And the process includes how you prepare the field, spread the donor material, spread the straw mulch, apply fertilizer for nutrients, and block the drainage ditches so that water does not escape out from it. However, we have certain challenges because this technique was developed specifically for restoration of bog peatlands, not the fen. And it was also designed for the weather in Quebec, which is much more wetter than Manitoba, that is drier. And it was applied to a land size of about 8 hectares, which is our own site is far bigger than what they applied it on. The question now is, since we want to restore to a fen, will this work for us? Since this design was basically for a bog pit, but like I stated earlier, the peatlands progresses from a fen base to a bog base. So there should be some fen materials that could be activated to see if this would work. Quebec is a much more wetter climate to Manitoba. We're having a different climate. We don't know if this will work, but it's worth to try. While, like I said earlier, our site is bigger, about 10 times larger than the 8 hectares. This is a little picture to depict what I've been talking about with the evaporation, transpiration data. We have higher rates of evaporation, transpiration compared to what Quebec has. Precipitation is about 580. Quebec has 960, which is wetter. And this is very important to what we are doing. So now this is the approach that I'll be taking to carry out this research. We have this area delineated in yellow, where we'll be applying furrow diking to make sure the water. We'll also be applying straw mulch in these brownish zones. And in all of this, we'll be testing the water retention strategies. And like I said, straw mulch will be applied, donor materials and fertilizer, just like we had in Quebec. This will be applied. We will also check various regimes of moisture, since the whole place cannot be said to have the same moisture content. We will need to monitor this, and we'll be creating an ecotone also to see how there will be an exchange of nutrients and water between the surrounding body and the peatland we are trying to restore. So my research question is that will this method work for our fen-based restoration process? And if furrow diking Will it help to retain the water or recreate sufficient surface variation to keep moisture levels higher? And will the ecotone will be constructing if it will be communicative between these two bodies around? And these are the data that will be collected to see if what we are looking at will be a success or we will need to modify our approach. Soil moisture content, we'll be monitoring the water table and groundwater movement. We'll also be setting up a weather station to take these parameters for study. And in summary, I've just told you how wetland plays an important role in the ecosystem of the world, and hence there is need to actively restore every disturbed peatland. So our study using the moss layer transfer technique, which has worked to transfer or rather transform a bog-based, returning it to what it used to be, is to see if it will work for our own peatlands with the addition of an ecotone with it. Thank you. And this is to thank my partners and funders for this research. Thank you.
This might not be a long question. Just so I get this right, you take like donor material and place it into the new area that you're going to, right?
Yeah.
Where does that come from?
Okay, like when you carry out an experiment, in my elementary biology, you have an experiment and you have a control, kind of. So now we have donor materials from an unimpacted site that has not been disturbed, and we have an impacted site that has been disturbed. So in our restoration process, we take these materials, those species of mosses and those species of sphagnum moss that is in the undisturbed place, we take samples from them and transplant in where have been disturbed, while we monitor to see how it will thrive and what will be needed for it to properly thrive there.
Okay, so yeah, basically what I was wondering is that like from like as nearby as possible, is that where that's coming
Yes,
from?
it is.
Okay.
It is.
Yeah, so oftentimes there's still natural peatlands surrounding the extracted area that they can just go to, because the less distance you have to transport, the better. And it's about a 10-to-1. So if you go into a natural area and take about an inch off the surface for a hectare, you can spread that over 10 hectares of restored area. And within about 5 years, you can't even tell you went into the donor area. It's like completely recovered, like you were never there.
Thanks, Pete.
Either Pete or Gladys, whoever can answer. So preferentially, you see that this kind of moss can retain more water, so they are more preferred for water retention, or you don't care what kind of diversity you have on the donor. You just take some piece and put it in your planned one, correct?
Okay, so when you're talking about species, already you have some surroundings from the donor. So we just tend to look at what we have already and not go bring in something from somewhere else that has nothing to do with that environment. So we just take from what is within that environment and see how we can put it. Now, when you talk about the water need of it, we can't have too much water. We need to make sure it's not too wet and it's not too dry. I don't know if that answers your question.
The diversity of moss species in Manitoba, Terry's been coming out to the field sites for 5 or 6 years now, and just recently discovered a 7th species. So there's not a lot of variety here. That actually is kind of a consequence of the bogs being quite acidic, that you actually decrease in species richness in bogs. But there is one species that we need to look for in donor sites. It's called Plectricum strictum. It's a pioneer species that grows very quickly and helps hold the soil together to prevent frost heaving. And with that present, it just helps stabilize the soil early on. And it's, I mean, even me, who can barely identify a deciduous from a coniferous tree in the middle of winter, can find that plant very easily.
Okay, I think we're going to move on to the next presenter. So our next presenter is Gladys Kibera. she's going to present on fen restoration in southeastern Manitoba, the role of ecotones. We heard Collins talk about ecotones, but now you're going to study ecotones. So Gladys obtained a Bachelor of Science in Environmental Studies from Masai Mara University in Kenya. And previously, Gladys worked with Green Ops Consultants in Kenya as an associate environmental expert specialist in sanitation management. So it's all yours.
Okay, thank you. On to series 2 of fen restoration in southeastern Manitoba. Before I get into ecotone, just so we are on the same wavelength, I want you to imagine you're standing in between two different environments. Like on one side it's forested, on the other side it's a grassland. Where you're standing now, imaginarily, that's an ecotone. It's an area of transition. It's in between. Now we'll move from area of knowns to unknowns. Known is wetland, to unknown is ecotone. So aim of my research is, in regards to fen restoration, is to determine the ecohydrological functioning of an ecotone that we shall be creating. Some of the research questions that I'll be looking to answer include, but not limited to, how groundwater varies around the ecotone, the relationship between soil permeability and retention rate also around the ecotone, also the nutrient cycling variation, because the ecotone is not flat, and then also with the alteration of water tables, we'll realize nutrients flows in at different rates, affecting how the fen will perform. Lastly, it will be to what extent do ecotones complement the fen restoration process. So what is a wetland? Like Collins mentioned, a wetland is any aquatic ecosystem that has water and allows for growth of plants and adaptability of soils and animals. So let me go back. Okay, into global statistics of wetland globally. All 20% of wetlands are actually found in Canada. That means you're very lucky. And 90% of these wetlands are actually peatlands. Now, according to the Canadian wetlands scientists who study wetlands in Canada, we have two types of wetlands, that is mineral-based and organic-based. For mineral-based wetlands, they are mineral because their substrate soil have minerals. Whereas for peatlands, they have peat, dead organic matter. So examples of minerals, we have open, shallow water marshes and some swamps. Now swamps, you can see it's in between, can be mineral-like or organic-like. And then we have fens and bogs as examples of peatlands. Some of the functions of wetlands include control of soil erosion and flood erosion, recreation, migratory areas for fish and birds, habitat for amphibians, source of food. Back in Kenya, we go fishing, and a source of underground water supply. They feed aquifers from underground. Also source of clean water. I'll give reference to my country and my home. My grandmother still fetches water from the nearby lake. So what is the importance of peatland? Why are we giving it so much importance now? So peatlands act as carbon sinks, and half of what, like I mentioned before, half of world's wetlands are actually peatlands, and this account for 3% of the total land surface. A healthy peatland absorbs three times as much carbon from the atmosphere as 3 times as much carbon from the atmosphere compared to forest. Forests can look as if they are big because, I mean, they are high and they are not as continuous as peatlands, as in forests have spaces, whereas for peatlands, they are continuous. That's why they hold as much carbon as possible. So differences between bogs and fen. Bogs, like stated, have acidic characteristics, and their pH varies from 3 to 4, whereas fens can be both alkaline or acidic. Bogs also are fed from the ground. Fens are fed from the ground or underground. Another difference is bogs have water tables of up to 50 centimeters, whereas for fens, the water tables are slightly constant. Bogs are low in nutrient and oxygen because of their acidity, whereas for fens, they have low to high nutrients again because they tend to vary between alkalinity and being acidic. So what leads to disturbance of peatland? By disturbance, I mean what leads to them being degraded. So we have agriculture where we clear the land to provide room for agricultural practices, forestry where we cut coniferous trees from the peatlands for firewood and timber for furniture, peat extraction. Now, peat extraction is our point of reference in our research because the land we are working on was degraded because of peat extraction by sun grower. Also, peat fires lead to degradation and climate change. This here is an aerial photo of my site, Elma, and it is, of course, it's a peatland, but now it is not performing its function of absorbing carbon from the atmosphere, but rather it is releasing carbon. So it is doing the reverse. Why? Because this black thing here is carbon, and chemists in the room will tell us carbon plus oxygen, C plus O2 equals CO2. So, and that oxygen is then, and that carbon is the CO2 is then released into the atmosphere, combines with other greenhouse gases, forms a blanket, and that leads to global warming and climate change. Now, this is what we want to reverse. So what's new in this research? Like Collins mentioned, we're going to use this technique, most layer transfer technique, and then we are restoring a fen and not a bog like in previous experiments. So what happened is during the extraction of peat, like I mentioned from our side, Sungrow, when they were extracting, they got to where the characteristics of the land were fen-like and not bog-like. So it makes sense for us to restore that piece of land as fen and not as a bog. Another new thing is that this is the largest restoration site in Southeastern Manitoba because, again, we are restoring a large size of land. It is 80 hectares. Under normal experiments, because of time and because this is a very expensive exercise, so mostly they will restore small portions of land, say 10 hectares or something like that. But us, we are dealing with 80 hectares out of 800 hectares of the whole Elma. And also our point of interest in the next 5 minutes will be ecotones. So like I mentioned before, ecotone is a transition area. It's in between that allows for movement of energy, water, and nutrients from high concentrated area, which is the natural ecosystem, to the degraded ecosystem. This is also an aerial view of my site, Elma. This is the forested bog. This is the natural ecosystem. This here is the ecotone, and this is where the fen will be restored. Now, to most layer transfer technique, we'll be getting mosses from here and elsewhere to here to replant here. From this aerial view, we can tell the characteristics of an ecotone physically before even getting to the site. So it is in between two different ecosystems. So most definitely its species, both flora and fauna, will be different. And then it's an area of tension because there's pull and pull of all these resources. And then ours is constructed. That should tell us we have natural ecotones. So moving forward, characteristics of ecotones, like I mentioned, can be natural or man-made. Ours is man-made, maybe narrow or wide. This is relative depending on what narrow is and what wide is, and also depending on where the restoration is taking place. And then species can be entirely different. So into ecotones, now that we've started to the unknowns, we want to know about ecotones. So some of the functionings of an ecotone, we derive its functioning from its uses, its importance. So it's water, its nutrients, and its energy. So we'll be getting to understand the dynamics, water dynamics of ecotone in relation to fen. We'll be learning the soil characteristics, soil moisture content, and soil permeability if water can be allowed to move through the ecotone into the fen as we do the restoration. Also, nutrient cycle, the ecotone has plants. It's a pit. So when water sinks in, will it allow nutrients to move into the fen to allow for growth of mosses and also vegetation patterns? This is purely for biologists. So another aerial photo of my site. Now, this is a map representation of Elma. This is the fen where Collins will be working on. This is the dark part is the bog. So Gladys Kibera, myself, I will be working on the red marked zones. This is the ecotone, and where I'll be carrying out my treatment is on the blue zones. Now, these treatments will help us determine how long it will take for this fen to be restored. So some of the treatments that will be carrying out will be slope transmissivity, will be altering the water table. Also will be carrying out soil moisture experiments, will be determining pit depth, and also will be placing tensiometers around the ecotone. This is what I mean. So this is the current water table. We will alter it to this new water table. So this is the piece of land, and then this is the new one. We will plant wells. Wells are just to supply water into this space, and then we will plant, we will have other experimental tools, tensiometers. This is a pit bucket to measure evapotranspiration rate. And then this is one site. We'll have this replicated on 6 zones on the ecotone. This here is Alex. This is Alex, our research assistant, and what I was explaining is this. This is a bucket for evapotranspiration. This is one of the wells of all that are here. Like I said, we have 6 zones, and then inside here we have the piezometer and the tensiometer to measure hydraulic height and to measure water retention. So knowledge gaps of this research, like I said, it is what we shall be doing. And now with ecotones, there is very limited information, either in literature or actual experiments, because it's a new technique. And so we are looking to see that with this experiment, it will be a success. And moving forward, scientists like us will adapt it into the restoration process. Also, in summary, I want to, wow. Oh, okay. So in summary, this research will help us to realize the importance of ecotones, because in most experiments, under normal circumstances, when there's restoration, we come from the natural habitat straight into the degraded habitat. We do not consider the land in between. So the newly regenerated environment is almost in shock. There is no time to allow it adapt into the environment. So we are hoping to understand this in future. I want to acknowledge my funders and my colleagues at MELS department, and also in-kind support from Sangro and funding from Whittington. That is a photo of us at Elma site in October this year.
Thank you.
Very nice presentation. So you can breathe a little bit now. At the beginning, you said about the pH change, right? That how one is acidic, more acidic than the other one. I'm just wondering, can you explain how does the pH is changing in a peatland when you are giving us the example of carbon emission and retention?
Okay, so now pH is in relation to microorganisms found in that system. Okay, so wow. Pete, please help me.
So eventually with the bogs, they become hydrologically isolated. So they're basically only fed by rainwater, which is what, pH 6 or so. And then just chemical properties within the peat and the peat itself just further exacerbate that, creating a lower pH. With fens, because they can receive surface water inflows or groundwater inflows, they can become in contact with like calcium rich clays and things like that have much higher pHs. You can have this huge range in pHs of fens. And so if you're harvesting the peat from bog peat eventually down into fen peat, because the fen gave way to the bog, like the bog succeeded from the fen, then essentially what we're testing or seeing is, does it make sense to try to target a fen ecosystem for restoration versus a bog ecosystem for restoration, because the chemistry of the peat left behind is more basic or has a higher electrical conductivity, which might be better for, or prove more successful to try to restore to a fen, basically.
She actually told us about the CO2 example. I was trying to drag her over there. Is the CO2 making it more acid?
Yes, it is. I mean,
Dissolved CO2.
I wasn't sure if that is what I want to say.
I look scary. Trust me, I'm not.
So maybe to add to that, I think it is also when plants die and microorganisms, they release carbon. So that is what is making this particular part of the soil to be acidic. And now fens succeed bogs. So that means what is up here is a bog. That is why it is acidic, because of decomposition of these microorganisms. Now, when you go under, I mean, when you move below, then this acidity starts to become less acidic. And then we get into the alkaline nature of the soil.
Perhaps maybe more of a comment than a question, but kind of connected with Mishimi because we come from biology. Yeah, so I'm something to think about and consider is then how does the biotic community change? Because I know that, you know, there are some differences in terms of fish and amphibians and what pH they can tolerate. So more of a comment than a question, but something that might be interesting to consider. I assume that's out of the scope of your project. You're going to be focusing on the peat side as opposed to the frog side. Sorry, that was a question. Are you just focusing on the peatlands as opposed to?
I am focusing on, sorry, I'm focusing on the peatland. We have a biologist from University of Waterloo who I'll be working with here.
Oh, nice. Excellent. That'll be really interesting. Yeah, thank you.
So we're going to move on to the last presentation for this afternoon, and this will be Akwasi Oduro. And we're going to, Akwasi is going to talk about mining atmospheric CO2, peatland restoration in a smelter damaged ecosystem. So maybe that'll get towards some of the things you were asking about, Sumi. Akwasi completed an undergraduate Bachelor of Science degree in meteorology and climate at Kwame Nkrumah, did I say that? Yeah. University of Science and Technology at Kumasi, Ghana. Akwasi served as a junior programs coordinator at the Institute of Sustainable Energy and Environmental Solutions, also in Ghana. So it's all yours.
Okay. So thank you very much for sitting through to the last talk. I know you may be wondering that we've had the first two presentations on peatland, why a third? So the importance of peatland cannot be overemphasized, just like the other presenters, my colleagues Gladys and Colin stated how important it is. And also, I would want to take this opportunity to acknowledge the works of Canadian scientists, especially my supervisor, Pete Whittington, for spearheading the charge to research into this important ecosystem and actually setting a mark for all global scientists to follow as well. So what my project focuses on is to work on a peatland restoration in a smelter damaged ecosystem. So right behind this picture background is a drone shot of Sudbury, where my field site will be located. And up on top is the super stack, one of the longest chimneys for smelter operations in Sudbury. And this is also a peatland. And you can look at the ecosystem, the fact that it has vegetation, the Canadian Shield, the wetlands, and all those stuff. I would zoom into more details in the project when I'm going. So the aim of my project is to develop a novel peatland restoration technique to address the impacts of industrial contamination and liming and determine modifications to upland regrading efforts to enhance landscape carbon sequestration potential. So it's more or less like a twin problem. We are looking at industrial contamination and liming as well. Now, as I zoom in, permit me once again to reiterate some bases. What wetland is, the fact that it's a land that is saturated with water long enough to promote aquatic processes. And this is from the National Wetlands Working Group. And the fact that Canada hosts 20% of the world's wetland and 14% of its landmass are also wetlands. And out of the 14%, 90% of them are peatlands. So that is why we are really focused on peatland, because if you look at the composition of wetlands in Canada, 90% of them are peatlands. So Canada's peatlands accounts for the largest proportion of global peatland carbon stock. That's the need for us to embark on this research. Now, what then is a peatland? So just like Colin said earlier, a peatland is basically an organic wetland with a peat accumulation of more than 40 centimeters. So there are two types. We have the bogs, we have the fens, the bogs being umbrogenous, the fact that they are precipitation fed only, and then they are very acidic. And then the fens being mineralogenous, the fact that they can receive rich base groundwater or surface water, and they range between acidic and basic. So what problem we are encountering is the fact that across the world, these ecosystems are being damaged at scales and intensities varied from whatever causes, such as climate change, resource extraction, altered land use, and contamination. So this is a similar story with my research sites in Sudbury. So this is a drone shot taken, I think, in summer this year of how Sudbury looks like. So you can now once again see the vegetation, you can see the peats and all the other parts. But interestingly, in the 1970s, this is how Sudbury looked like. So because of the smelting activities, now Sudbury is known for nickel mining. Unfortunately, it had an environmental impact on the area, whereby most of the large areas were devoid of vegetation, and you could barely see plants grow or green trees grow in Sudbury. So the city of Greater Sudbury decided to begin a regreening program, and this year marks 45 years since that regreening program started, and then they've been able to plant more than 3,500 hectares of land. And in the process of doing the regreening, they use lime and they grass it just to make sure they neutralize the soil and give it a very suitable condition for the trees to grow. So after 45 years, this is another picture of the same street taken in August 2023. You can now see the difference of how Sudbury is green now and how it used to be the same. So for the avoidance of doubt, this red triangles just indicate that it's the same street because you can see the piles and everything there. Now back to the picture I showed you earlier. So the problem we are confronted with is the fact that now from the chimney and the smelting activities, we have atmospheric pollutants settling on the ecosystem, thereby altering the importance and role of the peatland. Also, due to the regreening efforts, we find the liming moving from the uplands into the peats, also altering the peat. So we decided to carry out, and this was a colleague from another university, decided to carry out a preliminary research on the state of the peatland before we began the research. So this data is very, very complex. So I decided to break it down. What we did was that we picked 2 sites in the area, one on the step and one the step, and decided to check from 45 centimeters how old the peat was. So from the on the step side, realized that from 45 centimeters, the peat was 150 years old. Now, when we did same for my site, which has been the step, and at that same depth, we realized that the peat was 2,500 years old. So what this simply means is that we've lost about 2,350 years of peat, of carbon back into the atmosphere, which is very, very, very significant. That's the need for us to move into this site and at least experiment 1 or 2 things just to restore it. Now, so this is my study site, which I first visited during the reading break. So I just decided to take a live video. So you can see the trees that were planted. You can see the saturated water. And the fact that this site, unlike the other sites my colleagues described, Collins and Gladys described, this hasn't been harvested. But unfortunately, due to the atmospheric deposition of contaminants, you can find that literally it can't function as a peat anymore. So what we'll be doing is that we are going to experiment the already known technique we have, which once again, my colleagues explained, the MELS layer transfer technique, whereby we prepare the peat field, we create pools, which is optional. We gather plant material from the borrow area. We spread the plant material, we spread the mulch, add phosphate fertilizer, block the drainage, and then we are good to go. So these are my research questions. So just like I said, unlike the other sites, these sites have not been harvested. So literally, they haven't been, the moss hasn't been removed. So we are trying to see what novel peatland restoration technique that can be developed to address the impact of industrial contamination. So the already technique we know helps us address peatlands that are contaminated due to harvesting and all those stuff. So what modifications can then be done to find a novel way of addressing these impacts? Also, just like I said, the twin problem, we have the liming or regreening efforts also having a disadvantage of to our peatland role. So we want to know what modification we can also do to the upland regreening efforts to enhance carbon sequestration potential in Sudbury. Because regreening the place is also a good thing, but one way or the other, we need to also balance it there. So finally, my research question, the last research question will be, can the MELS layer transfer technique, which is already a known technique we know, then be adapted to help new MELS establish and bypass the chemically damaged material? So this is a Google photo of my field site and where the treatments will go. So we plan to apply 3 treatments on the site. So we have treatment 1, treatment 2, and treatment 3. So for the first treatment, we will remove all the ericaceous shrubs. So if you looked at the video I showed earlier, you realize that the field had all these shrubs. We need to then take a machine in, which currently is a bit of a challenge because where our field site is located, it's kind of difficult to move these machines into. So we are currently exploring options on how we can get that done. It was part of what I was supposed to do during the reading break, but unfortunately, it couldn't be done. Now, we also plan to add the MELS donor material on all 3 treatments. Also, add the straw mulch and then the phosphate fertilizer. So these 4 basically would be the MELS layer transfer technique being applied on all 3 treatments. Now, this is a shot of the field site of my other colleagues when these treatments were being applied. The straw mulch, you can see the moss layer already and the color contrast between the small mulch. The fact that this straw mulch has a higher albedo as compared to the other things. So now here is the difference. Now we plan to till the surface of the first treatment and not of the second and the third. Now tilling the surface will then give us or break the surface and make sure it is ready for us to apply the treatment. Now we decide to also add a buffer peat just to give us an optimal pH level on the third treatment and not for the first or second. So what we are doing is that we are trying to see what the significant difference will be with tilting and adding the buffer peat. Tilting and adding the buffer peat, and what the outcome will be would inform us on the decisions to take. So basically, this is what I explained. So this is my field site. Now we intend to create these dimensions about 15 times 50 meters with a 5-meter buffer between them. So we have the first treatment, the second treatment, and the third treatment. Now what we do is that we'll then be measuring the soil water, the soil moisture, and all the necessary properties we'll need. And based on that data, it would advise us on what approach to do. And I'm sure when we gather here once again, we'll have enough data to tell you the approach you should take. So I would like to express my sincere gratitude to the National Science and Engineering Research Council, Alliance Mission Grants for funding this project, and to my supervisor, Pete Whittington, Peter Bucket, who also spearheaded the regreening efforts in Sudbury, and my colleagues for their invaluable contribution to the project. These are my references. Thank you.
Just before I duck out, it's probably important to note that there's quite a few other authors on the grant that we got for this. We thought the news was going to be announced a couple of weeks ago. We found out back in the spring, but it's still embargoed. So there's been no announcement of the grant, even though we've been doing work for 8 months now.
A very nice talk. One question. So you mentioned that nickel mining has a profound effect on the areas that you showed in the picture around Sudbury. I'm sure that wasn't nickel that was spilled. So could you give us a few words about how those things affect, how the mining affects in general? Like what is the material that affects over the next few years?
Yeah, so during smelting activities or nickel mining operations, you realize that during the processes, there is release of sulfur dioxide into the atmosphere. So these atmospheric emissions, they said, settles on the peatland, then altering the chemistry of it. This is more or less like an interdisciplinary approach. So we have other colleagues and students from other students looking at the other areas of it. So basically that is what has been affecting it. But one way or the other, these nickel mining companies are also doing their bit in making sure that they release the pollution they create. So now they are climate smart technologies they're employing, and the fact that they even collaborate with researchers, give them funding and all those stuff just to make sure we learn new ways to save our environment. Yeah.
And another question, what happens when you spell peat as P-E-T as opposed to
what happens when you spell peat as P-E-T-E?
Yeah, instead of P-E-A-T, what if you spelled it P-E-T?
I guess you get a kick in the butt.
Nice. No, actually, we're going to go online next.
Yeah, a question again from Michael. Just being curious, how much depth of peat loss would have resulted into 2,350 years of carbon return to the atmosphere using the 245 centimeter depth images alone? That's roughly 705 centimeters, 7 meters. Are there other stuffs at play? I might be getting this wrong. There's a lot of questions involved in that, it seems. So. Yeah. So, yeah, just some, I don't know if you have any thoughts with his sort of comment. I think he's doing some rough math there, but yeah.
Oh, okay. So just I think his question will definitely be related to the part where I showed the two peatlands, the stepped and the understepped. So basically what we found out was what I put out. The fact that on the understepped peatland, the peats at a certain depth equal to the step peatland were actually different. I mean, this was just 150 years and that was the 3,000 and something. Yeah, sorry.
Who had their hand up over?
Gosh. Every time I hear about peatlands, I'm always like, wow, these are really, really cool. And I forget about them then. But what I saw when you were outlining those areas was an enormous amount of work. Because I've been through those mossy, boggy grounds and it's hard to move. So how many hours goes into those 3 treatments do you anticipate?
Well, it depends. I mean, I can't specifically say the number of hours, but we are going to be on the field. I mean, we'd have our first real field season in summer, in 2024. So during that period, we hope to have, I mean, carry out almost the basis I said. Yeah.
Quite a few weeks then.
Well, this is the problem is that these, because they're still natural, air quotes, there isn't the infrastructure to get big tractors and trucks and tillers and stuff in. So we're trying to make these treatments small enough that we can hand bomb these things in. Like we figure we need to move about 75 cubic meters of moss. So whether it's helicopter or snowmobile or ATV, like the logistics of these projects are going to be enormous. And so this grant that we got, which was an NSERC Alliance missions grant, required no money from industry. So we got $1.5 million with no money from industry for 3 years. We're using this to test some ideas to then go to industry and get a much larger grant for 5 years where they have to pony up some money. But the logistics of it, like, it might be dozens of helicopter hours just moving stuff from the parking lot. Like what Akwasi didn't show is that one of those sites is 200 meters from a road. So it's really close, but it's a 1.5 foot wide trail. You can't drive a pickup truck on it. So yeah, I mean, that's going to be a real issue.
Why do you have to remove the shrubs? Is that just to get the equipment in there later, or?
We just don't think we're going to get any contact with the ground. It would be like spreading anything over top of a forest and trying to get it to hit the ground underneath. If it was important to put the material on the ground underneath the forest, you'd have to cut down the trees.
So does it make any difference to your result if those shrubs that would be part of the ecosystem and the nutrient cycling and aren't going to be there?
Yeah, so they actually play an important role sort of hydrologically anyway, but we figured by just removing the tops of them, we can put the stuff down, and then over a summer or two, you'll start to see a canopy of them form back again. That's also part of the experimental design, was that we were just going to clear cut it because we don't think we can put anything down onto the ground underneath it. So we're just going to be spreading donor moss, which is just going to be hanging in the shrubs dying, right? But we reckon the acacia will grow back quickly enough that they can start to have a shading impact on the surface, which helps with minimizing evaporative losses.
Okay, last question to Masumi.
Pete, last question.
It's not question, it's congratulations for the NSERC Alliance grant, and he's like, right now I'm giving something positive, he's living, but good for that. I'm just wondering, like, when you say that is treatment neither, is that your control when you are comparing that to your treatment? What is your control in the experiment? I'm trying to understand.
What is my control in
when you are saying treatment 1, treatment 2, treatment 3, I'm assuming between 2 treatment, is that your control, or the one you say treatment 2, neither, that's your control?
So between the 2 treatments, that's the control. That's where we have the buffer. These are the control.
Okay.
Yeah, but the difference between the treatment 2 is the fact that, you see, we are doing tilling here. The treatment 3, we are adding a buffer pit, but the treatment 2, we wouldn't be doing tilling. We wouldn't be adding any buffer. Then from what we will get, it would advise us on what needs to do. So the control is just between the buffer. I mean, the treatment.
Okay, I think, I guess, are you, yeah, I think I'll allow Dr. McGonigal to ask a question and then we'll wrap up.
So you're going to apply these treatments. What are you going to measure subsequently? What will be your response variables that you'll measure to see the impact of the treatments?
Yeah. So we already have, if you look at the picture, we have our weather station. So on my part of the project, I will be measuring the water table just to know how, varying it is. We would be looking at the soil moisture. Yeah, basically, and some other hydraulic properties of this particular site. Yeah, so those are the things I've been.
Okay, thank you very much. And thank you everyone for attending today. I had a great bunch of talks and we look forward to seeing you guys in a year and a half time presenting what you found. So